All right, dear colleagues and dear friends, it's so sad that Erkki is not with us anymore. I have very good memories from him from the times when I was first recruited to Institute of Biotechnology by Mart Saarma. And at that time, there was this young Estonian guy also around, and together we did some experiments with 2,5-A, transformed plants that was really new at that time. I had spent some time in Belgium at in University of Ghent and learned how to do this thing. The story I'm going to tell you now in the next 20 minutes really sort of dates back to those times. At the same time I was moving to the institute, we had already collaboration with the Kemira company who was promoting plant biotechnology at that time. And it was actually Kemira who had bought a Dutch breeding company breeding roses and gerberas. That sort of brought gerbera into our field. And we fell in love with gerbera, and we're still working with gerbera, different aspects in it. We started by studying the color and trying to engineer the color of the flowers. And I mean, by color here, the water-soluble anthocyanins that give orange and red and blue and purple shades to many ornamental flowers, and of course wildflowers as well. So gerbera comes in orange and red colors, the different varieties, but there is no blue colors in gerbera. And this pathway to the colored anthocyanins branches actually into 3 parts, and one of them is blue. And gerbera simply, like roses, don't have the blue branch. But now we're talking about orange particularly. And if you look at this biochemical pathway, you see at the branch point that actually making orange is the simplest thing of these. The only difference between those 3 branches is the hydroxyl group in the phenolic, in the aromatic ring upright. There is 1, 2, or 3 hydroxyl groups. And there are 2 enzymes that are adding these hydroxyl groups. So orange petunias don't exist in the wild or in the traditional breeders' collections. This picture is from the backyard of Institute of Biotechnology in Pitäjänmäki. I took it because it very nicely shows this phenomenon that there are only red petunias and blue petunias, of course white petunias without any color, different shades of these, but no orange ones. And why is this? Because it should be easiest to make orange. The reason is in substrate specificity of a particular enzyme, the hydroflavonol reductase here, after the branch point, and the pelargonidin DFRA enzyme doesn't accept dihydrogenferol as a substrate simply. That's why it doesn't make the orange color. But many other plants do, like gerbera does, maize does in its kernels, and already this information gives kind of a recipe of how you could actually fix petunia in making orange. You just have to transfer a gene from a suitable plant into petunia. Well, not just any petunia, but a petunia that has a mutation in those 2 hydroxylases, like this lab line RL01, which is a mutant for those hydroxylases and accumulates dihydrogenferol in its petals. It's nearly completely white. We were not the first ones to do this. This engineering of the metabolites was done by Peter Meyer in Cologne, Max Planck Cologne, a wonderful example of this logic, taking the maize DFR and putting that into the RL01 petunia, and you get orange pelargonidin formation in the plant. Like you see from this figure, they were kind of pale orange, completely new new color. So at that time, we were also using gene transfer to study our genes and to show function of the genes. And in Gerbera, we started from the color biosynthesis genes, and we of course did the same thing with the Gerbera gene, not now to sort of demonstrate a new thing, but to sort of demonstrate that the gene that we had isolated from Gerbera indeed was a DFR, because we could convert our L01 petunia into an orange flowered petunia. Actually, our gene worked better than the maize gene. We did also some studies of why this would be the case. It's not silenced in a similar way as the maize gene is. It's actually stronger in activity. But then at the same time, also breeders had taken this, these lines, the maize lines, under work. This line that we transformed and Peter Meyer transformed is a lab line. It's not a garden petunia. It's not particularly showy. It's an inbred line, very good in the lab and so on. But then breeders in Holland, in this small breeding company, which is nowadays part of Syngenta, took the Cologne lines and crossed it with garden petunias, with big flowers, with intensive colors. And they were able to breed this poor little lab petunia with the color into really brilliant, wonderful orange petunias. The last sentences of their paper in Euphytica ends like this, that this transgene behaves normally in all tested aspects. Of course, that's what we always expected, that the genes that we put into plants, they behave normally. And they tell that it's been successfully used in their breeding programs that aim at developing commercial F1 varieties with this orange color trait in petunia. But look at the year. We are now 1995. The gene technology law in Europe had just been sort of built. Actually, in Finland in 1995, it came into an effect. And what do you think when you're reading this paper in 1995? That yes, this would be great, but I think this is just sort of wishful thinking. So very good idea, like many good ideas in genetic engineering, are practically then stopped by the legislation because it's simply too expensive to get to the market with GM traits, and that's why only the biggest companies with the biggest crops are doing it still today. So, end of story. Then 20 years passed. This is railway station in Helsinki, 2015. I was walking on the railway station on my way to Viikki, to the lab in Viikki campus. And I saw these petunia flowers on a box there on the railway station. And I thought that, oh, wonderful colored petunia. How is this possible? This must be some kind of a mixture of red and maybe yellow carotenoids, but the plant is really doing it beautifully. It's really nice. And of course, I picked the flower to my backpack.
Olen praegu konverentsil.
And I guess there was nothing. Okay, I picked the flower in my backpack, and I had, of course, to run. Excuse me?
No.
Okay. I extracted the anthocyanins from this plant and ran HPLC, and suddenly it was not carotenoids on red, but this plant was really producing pelargonidin. So this is not possible, basically, historically. Was it still possible that the breeders had found a mutation that makes pelargonidin? Or was it possible that these actually are the transgenic lab lines that somehow had sort of seeped into the breeding programs? And by some PCRs, it was sort of easy to show that actually this line contained exactly the same genes as Peter Meyer's construct, shown here as a figure figure in nature. This was of course published soon biased, and this really sort of started a sort of a big mess. Here a journalist in the Science magazine is explaining, he phoned me and I gave an interview, is explaining how the carnage of petunias began, the bloodbath of petunias began, because none of these orange petunias were registered as GM, so they were basically all illegal, not only in Europe, but also in the US. And so the massacre started, and petunias carrying this gene, originally from Cologne, were destroyed. And if you look at this long list of petunia cultivars that were detected as illegal GMOs, there are orange ones, of course, but there are also other colors because breeders use each other's cultivars in generating their own cultivars. The list just sort of goes on and on and on. So now this was the end of the petunia story. They're not there anymore. But now new technologies are emerging. Gene editing technology. Would it be possible to use this here? The CRISPR-Cas system just got a Nobel Prize Tuesday this week. And of course, everybody, all molecular biologists have been sort of practicing and testing this system. Here you see CRISPR-Cas cutting the DNA. You can make easily loss of functional mutations. But what is wonderful, you can also tweak the DNA and edit some bases. Another question is that could we maybe edit the Petunia DFR gene, DFRA, into a pelargonidin producer? This is an old paper from a South Korean group by Gil-Chu Choi, where he was playing with our Gerbera gene that he asked to use in his experiments at the Petunia gene, and made some fusions of different combinations, and came to the conclusion that in this sort of stretch of amino acids that's shown there on the right, there are particular amino acids that are shared by those that will make pelargonidin, the orange color, that the Petunia doesn't have. They also made a mutation in the Gerbera gene, our gene, that changed N to L at position 134, and even more liked to make pelargonidin, the DHK as a substrate. So we took the Petunia gene and we made a couple of mutations like this D to N change, and then we changed it to L as well, according to what they had done, what Gil-Chu Choi had done with Gerbera. Some of these changes require a couple of nucleotides, but this one particularly could be actually done by G to A editing. So we haven't edited the Petunia, still practicing that part, but we made these mutations and we tested their substrate specificities in Pentamiana by agroinfiltration, a technique where you're infiltrating agrobacterium into particularly Pentamiana tobacco, something that emerged then in the 2000s, and something that I always thought, why didn't we think earlier about this in the 80s? Because it's such a simple technique. We used a wonderful hypertranslatable vector from George Lomonosov's lab, and here you see what our mutations are making. So on the left, there is the wild type DFRA of Petunia. It doesn't like to make pelargonidin. When we make this D to N change, that is in many other plants, doesn't change a lot. But the D to L change actually changes the substrate specificity upside down. It likes pelargonidin. It likes DHK that makes pelargonidin. So the idea here would be to sort of just tweak a little the Petunia's own gene, and this way make a sort of orange Petunia that would not have any foreign genes, just an amino acid change, just a couple of nucleotides changed. And the scientists all the time, when this method was developing, thought that this, of course, is not GM because we don't put any foreign DNA in. It can be done just with the Cas9 protein, for example. To everybody's surprised. Then 2 years ago, the Court of Justice of the EU decided that although mutagenesis is exempted from the gene technology law, the new methods of mutagenesis, this is the interpretation of the judge, are not exempted. So the edited versions of natural genes are GM at the moment in Europe. So some people think that this is now the end of the breeding innovation in Europe, maybe a bit too dramatic. But anyway, it was put a stop to sort of lots of hopes in improving plants with these new techniques. Interestingly, elsewhere, like in US, they are going the opposite direction. And the USDA is declaring that single base pair substitutions, you don't even have to ask. They are not GMOs, or deletions of any size, or insertions from plants that are crossable, like related species. And elsewhere, similar developments are going on. Europe is kind of alone, this red region. Well, not alone, there is New Zealand. And EU has decided that, okay, let's look into it. And they have been sort of asking for clarification and for a sort of next level of decisions. And this document should be handed next March. But let's see what happens. EU has been very sort of illogical in this sense. Maybe we are sort of stuck to this interpretation also in the future, although I sincerely hope not. I will finish here just by thanking my postdoc Annie Pasandi and a number of students in the lab with which we have been sort of developing the assay and doing the mutations. I will stop here. If we have questions, not sure, maybe I can ask them. Thank you very much.
Thank you very much, Teemu, for your wonderful presentation. And I'm really happy that you have risen the issue about legislation of GMOs and edited plants, because this is really important. And I think that all plant scientists today, we should do really a very loud voice. This would also be what Erki would do in order to have a change. And really next March is a very important date for us. So let's hope for the best, and let's hope that edited plants will not be legislated finally in Europe as GMOs, because this is just a tragedy. Because of technical solutions, it's not so easy to ask you, but I myself, I just want to ask one specific question about the editing you did. So you were referring to a substitution of one nucleotide, and you used CRISPR-Cas for this. I was just curious to know exactly which kind of CRISPR-Cas system, because I mean, you were not doing just an induction, but something different, because it's a substitution of a nucleotide.
Unfortunately, we are not that far. CRISPR-Cas is sort of easy on paper, but when you start to practice it, it's not that simple. So what I showed you were in vitro generated mutations. And that, of course, is the first step. We would like to see how it works. But the idea is to use, well, there are these deaminase-based editors. One of the mutations can be done with those. But what I'm really looking forward to, of course, is the prime editing, which on the other hand works in humans, but in plants there are not too many examples. We also have to sort of make we have to find a sort of a certain cultivar where this can be done, because it cannot have these hydroxylases and things like that. Mm-hmm. So I just wanted to share you at this point what we would like to do with this sort of colorful topic.
Mm-hmm. Okay, thank you, and good luck, and hope to have you soon in Tallinn when the situation.
It would be wonderful to come again.
Yes.
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 Okay.
Thank you. Thank you, Teemu. Bye.
Bye.
Bye.